Usually the main species I study are large whales. But some of the first marine mammal and cetacean work I did was on harbor porpoise, and it actually started in the late 1970s up in Glacier Bay, and harbor porpoise were one of the species we had up there. But for most of my early career working in Puget Sound, and I started research on marine mammals in the early 1970s when I came out here to go to Evergreen and Olympia. And as my undergraduate research project started studying harbor seals. But over the course of the many years, harbor porpoise were that species that used to be here and had disappeared out of Puget Sound. And kind of through my career, I've come back occasionally to harbor porpoise. And it's really exciting that we have some very new developments with harbor porpoise right now that are allowing me to do a little more work back on this species that I've kind of studied, I want to say at about 10-year intervals coming back to that. But mostly I spend my time off the West Coast studying blue whales, humpback whales, gray whales, fin whales are the much, much larger members of the cetacean families. Blue whales, the largest of all the cetaceans, and harbor porpoise, the smallest of all the ocean-going cetaceans. So it seems like I've spent time at either extreme on the cetacean size spectrum. There are a number of names of other people up here, and this is probably only a partial list of some of the people that have been involved in harbor porpoise research around Washington State and who I've stolen some of what I'm going to show you from. And I do tend to show a lot of figures and tables. First, a little bit of background. And you'll see a map in the lower left corner there, and everything outlined in blue there shows the range of the harbor porpoise. And you can see that it occurs in coastal nearshore waters of both the North Pacific and the North Atlantic, and extends down into more southern portions of the Atlantic. It goes into some of the inside waters. And it goes by different names in different areas. It's sometimes been referred to as the common porpoise in, for example, European waters. It used to also be, and if you've seen harbor porpoise, you'll understand why it's called this. It used to also have the very derogatory term of being called the puffing pig, was another name for it. But you can see that very coastal distribution for one thing, and that's one thing that characterizes harbor porpoise. They're very near shore animals, and that term harbor porpoise makes sense for that reason. They tend to be sort of hard to study because they are small. They, unlike some of the dolphins or Dall's porpoise that readily approach boats, they generally tend to avoid boats and will even move away from a boat and sound sources, and they don't seem to like echo sounders. They respond to that. They are pretty vulnerable to some human activities, and I'll touch on that, and that's part of the reason I got involved in studying them. Their size is only about 5 feet long. Typical weight is in the 100, 150 pound range, so not very big. They're dark on top and lighter colored underneath, but unlike killer whales or unlike Dall's porpoise, they don't have a sharp black-white delineation in color. It more fades from light to dark. And you just have trouble getting a good view of them. So unlike if I'm talking about humpback and blue whales, and I get to show nice dramatic pictures interspersed with my boring figures and tables, I don't quite have that luxury here. So there'll just be a few pictures because they're hard to photograph. They're tough animals to get close to. They have a pretty short, intense lifespan. They actually become sexually mature at about 3 years old. The average lifespan is only 10 or 15 years, so they're not as long-lived as many of the other cetaceans. And so those are some of their kind of basic characteristics. Now, one of the things that got us interested early on in harbor porpoise is they do seem to be highly vulnerable. They have a pretty long history of easily being caught in fishing nets, especially gill nets. And partly it's that coastal distribution. It's also that I think they become somewhat oblivious when they're feeding. Some of the areas that they're dense are often areas where there are gill net activities. And probably some other factor is the fact that they don't tend to react well to human activities. So in my early research in the 70s and 80s, we started to document the fact that harbor porpoise had largely disappeared from Puget Sound. We still had harbor porpoise up here in the San Juans and in the Strait of Juan de Fuca and offshore, but it was this dramatic change in Puget Sound that kind of characterized some of the early research I did, was trying to document how that had occurred. And it was interesting to look at that time. We looked at other areas like San Francisco Bay, the Baltic Sea, the Wadden Sea in Europe. These were areas that used to have large harbor porpoise populations, much like Puget Sound, where those populations were in trouble in those areas. So it seemed to be a little bit of a unifying theme around the range of harbor porpoise, that they had trouble where there was lots of fishing activities or heavy human activities. And I just grabbed this. If you can, the National Marine Fisheries Service maintains these what are called stock assessment reports that document the different fisheries that interact and are known to take harbor porpoise. And I won't bother going through that. Now, one of the early things that we became involved with, and because it was a critical issue, and it's often something that's underestimated in trying to kind of manage and protect marine mammal species, a critical element is understanding how the population is structured. And what I mean by that is often you have mortality in very specific areas, and you can estimate how dense harbor porpoise are and how many there are in a big area. But unless you understand to what degree there are distinct populations in different areas, you don't know whether to manage the whole giant area as one group and consider, a localized impact is going to be quickly replaced by animals from outside that area, or to what degree do they form these very regional-based populations. And so a lot of early research that I did was trying to tease out different ways that we could figure out to what degree was there movement of animals among regions, and to what degree are there these distinct populations. And for example, this is on the right, you see a map of the West Coast, and on the West Coast here of the US, harbor porpoise extend down to Point Conception, which is just north of the Southern California Bight and north of LA. And all of those kind of areas that you see separated by lines going up the coast are what are now recognized as distinct harbor porpoise populations. And I'll show you a little bit of the research that led to realize that it wasn't just one big population on the West Coast. So the earliest study, and you can sort of get a feeling it's an early study because it's a very crude map there. That's actually from the days where when you wanted to do a map, rather than do it in GIS, you would actually draw it and use press-on letters. So this is a map from those days to generate it. But what this comes from is in my earliest research on harbor seals, I had been studying contaminant levels, PCBs and DDTs. So we ended up looking at this in harbor porpoise and comparing the ratio of some contaminants, PCBs and DDT in particular, to each other, but also another compound called HCB, and looking at how it varied, because we knew these compounds were highly stable and they accumulated through the lifespan of the animal. And while you saw quite a bit of variation in depending on the age of an animal, how high the concentration might be, or its sex, the ratio tended to be fairly stable in a region. And when we studied this up and down the coast, what it actually revealed, and on this left side, you'll see little clusters of points. That's where harbor porpoise stranded, and we tested them. And these largely ended up showing highly distinct ratios of these very stable contaminants. And it right away told us that these were pretty distinct groups. If there was a lot of intermixing, you'd see much more overlap than we were seeing. And we had a couple of ways we could look at this. This is what's called a discriminant analysis. And basically those kind of polygons you see up there kind of encircle all the values of a particular group of harbor porpoise. And you can see that they tend to cluster differently. Now that was kind of our earliest effort. But now there have been some more sophisticated methods. And now this is an interesting study that unfortunately hasn't really quite seen the light of day. It made it into an unpublished report. But this is work that Brad Hansen did, where he actually captured harbor porpoise from up here in a net, because they're prone to entanglement, and attached VHF transmitters to their dorsal fins. And then recorded what kind of positions they got for these harbor porpoise. And what's shown here is the positions they got of some of the harbor porpoise, and there were 4 of them for which they had transmission durations of over 6 months. And each of these different colors are a different harbor porpoise. So bear in mind, each of these sets of points represents positions over greater than a 6-month period. But you'll notice here, all of these black points here encompass one harbor porpoise, and it was never seen outside this region in that 6-month period. And over here is another group in yellow. You have another one that varied a little bit widely from up here down to here, and then there was another one up here, north of Orcas Island that just stayed in that area. So over greater than 6-month period, this was a surprisingly minimal range of movement of these animals. So this also hinted at the fact these animals tend to form fairly localized feeding populations in different areas. And you do see movement, but it wasn't very large scale movement. And then the latest technique that has helped explore this is genetic techniques using something called mitochondrial DNA. And I think, let me just kind of cut to the chase. This is basically locations that harbor porpoise stranded and for which they could examine this genetic marker. And I won't bore you with this complicated figure other than to tell you that it revealed these significant genetic differences among most of those West Coast areas I showed you, and even some hint of separation within some of the inside waters. So these kind of stability of these units was enough that it showed up as a genetic marker. And you had different ways you could plot this, and I don't think I'm going to spend too much time on this. This is some of the divisions looking at California, Oregon, Washington, and how they were clustered. But here's another view of the results of the genetic results, kind of